So, hello everyone. I'm Megan Rippy, and I'm a postdoctoral researcher at the University of California, Irvine. And what I want to talk to you about today is really linking biofilter design to fecal indicator bacteria removal. So we're transitioning from nutrients to fecal indicators. And the real reason I want to talk about fecal indicator bacteria right now in sort of this environmental focused session is because meeting bacteriological water quality criteria has been cited as one of the primary barriers to implementing green infrastructure in urban systems where the end goals are things like human and environmental health. All right, so what exactly are the fecal indicator bacteria? Sort of moving on to slide 2. So fecal indicator bacteria are broadly classified as non-pathogenic enteric bacteria, meaning that they live in the guts of warm-blooded animals. And they tend to have high concentrations in feces, often exceeding 10 to the 7 bacteria per gram of fecal matter. And because they have these high concentrations in feces, they can be used as proxies for inputs of fecal pathogens in drinking and receiving waters. There are 4 major groups of the fecal indicator bacteria: the total coliforms, fecal coliforms, E. coli, and Enterococcus. The total coliforms are a group of gram-negative, rod-shaped bacteria that have both environmental and fecal constituents. The fecal coliforms are a subset of the total coliforms, and then E. coli is a species sort of within the fecal coliforms. The Enterococci are actually, or Enterococcus, are a genus of bacteria that are separate from the coliforms. They are gram-positive coccoid or round-shaped bacteria, and there tend to be about 50 species within that genus. One thing about Enterococcus, you see on slide 3, is that they tend to be the preferred proxy for pathogen inputs in marine receiving waters because they have the highest statistical correlation with human health in those marine waters, whereas E. coli, sort of on slide 4, are the preferred indicator in freshwater systems. So this means if we were designing a biofilter to meet criteria in freshwater systems, we'd be looking at E. coli, and if we also wanted it to meet criteria in marine systems, we'd have to add on Enterococcus. So a little bit about fecal indicator bacteria in practice, moving on to slide 5. One of the things I want to point out is that they're really imperfect proxies for human pathogens, and that's because they have non-human sources, things like animal feces, for instance, and also there are problems with environmental regrowth of these bacteria, which means that the relationship between the human pathogens and the fecal indicators is not consistent over time. But despite all of these difficulties, these bacteria remain our regulatory currency, reaffirmed as recently as 2012. So they're really relevant from a management perspective still. What exactly are our criteria? In the US, we have a variety of different criteria. We have ones for potable use, sort of drinking water criteria, as well as non-potable use, sort of our recreational water criteria. And for biofilters, probably looking more at the non-potable use criteria. And these come in sort of 2 flavors. You have your sort of statistical criteria, things like geometric mean standards and statistical threshold values, which basically say that a certain percentage of samples must have less than a given concentration of bacteria per month. And then you also have your single sample standards, things like the Beach action value, which just says in a single sample you must have less than a certain concentration of bacteria. So now I'd like to talk a little bit about fecal indicator bacteria in stormwater and dry weather runoff. So we're on slide 6, and compare those concentrations to some of those criteria, the non-potable criteria I was just talking about. And the first point is that fecal indicator bacteria concentrations in stormwater and dry weather runoff are often high. This is nicely illustrated by this table from Grevel et al. In 2013, and I should mention that all of the references are in the final slide of this talk if you're interested in any of this in more detail. And they show that the ranges for these fecal indicator bacteria are quite broad. They can get up to about 10 to the 6 bacteria per 100 mil. The medians are also quite high, and if we compare them to the Beach action values in red on the far right, we can see that the median values in stormwater can be 1 to 2 orders of magnitude higher than our water quality criteria. So this really means that sort of moving on to slide 7, that because our modern storm sewers tend to route stormwater away from urban centers and into urban receiving waters to prevent flooding, those receiving waters are often impacted by stormwater-associated contaminants like fecal indicator bacteria. And this is really nicely illustrated by Newport Bay, California. That's that image, the schematic on figure 7, which is actually, if you look at all the blue dots sort of around Newport Bay, blue and red, those are all storm drains. There's actually 219 storm drains that surround Newport Bay, And in a recent publication by myself and colleagues, we illustrate that dry weather discharge from those small storm drains is really a dominant contributor to shoreline fecal indicator bacteria contamination in the bay. So what this means, kind of moving on to slide 8, is that when we think about green infrastructure design, we really need to take fecal indicator bacteria into account. And to kind of hammer this point home, we heard in sort of earlier talk in an earlier session that the cost of redesigning urban water systems to meet water quality criteria and keeping sustainability in mind, things like green infrastructure approaches, is really high, more than $30 billion US just for Southern California. And this means that if we're going to be spending this kind of money, we really want to make sure we're dealing with all of our criteria. So that means including fecal indicator bacteria. So for the remainder of this talk, I want to look at this a little bit from a stormwater biofilter perspective and talk a bit about how we could be able to do that, I guess. So what exactly are stormwater biofilters? We've talked a little bit about this. Stan went over some of it quite nicely. So this is slide 9 now. And really, stormwater biofilters in general are just low-impact development or water-sensitive urban design systems, depending on where you're from, that are intended to remove suites of pollutants from stormwater or wastewater by physical, chemical, or biological means. They tend to be characterized by a small spatial footprint, so taking up about 2.5% of their catchment area, and they have vertical flow through layered media. So what this means is that water comes in sort of in an inflow pipe and enters a ponding zone, like Stan was illustrating. It percolates down vertically through layers of filter media until it enters a perforated pipe in the bottom of the system. So these are lined-in pipe systems I'm talking about. And then it can either flow straight out of the outflow, or in certain system designs, submerged zone systems, you can actually elevate the end of that outflow pipe, creating a saturated or submerged layer at the base of the biofilter that tends to have low oxygen and favor anaerobic microbial processes. Now, in addition to all of this, biofilters are also characterized by their biocomponent, so their vegetation, soil microbes, and animal communities, all of which contribute to the pollutant removal processes as stormwater moves through those biofilters. And sort of as you look at these schematics, it's obvious that there are a lot of different biofilter compartments or components. Your vegetation, you've got the ponding zone, filter media, submerged zone layers, all kinds of things. And so I'm going to argue kind of on slide 10 that if we want to be optimizing biofilters for fecal indicator bacteria removal, we really need to understand both the effects of individual biofilter components on removal and how these components work synergistically to impact removal, because the whole is more than the sum of its parts. But to start, we're going to look at these sort of individual biofilter components effects, and I'm going to talk about it from a very conceptual framework. If you want more details, all of this is presented in more detail in a recent publication of mine in Wires Water. So regarding the filter media compartment, filter media effects on FIB are largely associated with things like straining and physicochemical filtration, and that's because bacteria are really biocolloids. So they're 1 nanometer to 1 micrometer in size, and they're going to be impacted by the processes that affect other colloids. So what is straining? Well, straining is really where colloids become wedged at grain junctions. So in our little cartoon image here, that would be bacterial particle A, or stuck in narrow pore throats. That would be bacterial particle B. And physicochemical filtration is when colloids are colliding with sediment grains, which you can think of as collector particles. So that would be bacterial particle C. Or at the air-water interface in sort of an unsaturated system, that would be particle D. And then they're going to attach whenever the attractive forces are exceeding the repulsive forces. And there are a wide range of attractive and repulsive forces that are controlled by the bacteria themselves, by the grain collectors, by the ionic strength of the stormwater. It's more than we can get into at this point, but those are just things to keep in mind when thinking about these systems. So moving on to slide 12, we can think about our vegetation compartment. And vegetation could impact fecal indicator bacteria through things like root adsorption and exudate release. So for root adsorption, it's what it sounds like. You're just having your fecal indicator bacteria or your biocolloids directly attaching to roots. That would be particle A. And exudate release occurs because some of these roots are quite leaky. So they can release, in some cases, harmful compounds to bacteria, sort of antimicrobial compounds. But they can also release things like nutrients or other stimulatory compounds. And this is a process called priming. And we don't really know whether or not fecal indicator bacteria are going to be stimulated, so that would be particle B, or inhibited, or a dashed B particle by root exudates. And that's something that people are still looking at. So moving on to our submerged zone compartment on slide 13, submerged zones could impact fecal indicator bacteria removal by a variety of means, things related to competition, biofilm formation, or predation. So looking at competition for a bit, the submerged zone, because it increases saturation, it has the potential to connect microbial communities that would be disconnected in a more unsaturated system, which can increase competition. And it's not totally certain what this will do to fecal indicator bacteria themselves, and that's what's shown in sort of our little particles A here. Some of them are dashed, indicating they're dying, and some of them have a solid boundary, indicating that they're doing just fine. And it's likely to depend on the communities that are interacting, that are competing. Similarly, when we look at biofilm formation, it's possible that the increased moisture in submerged zone systems could lead to more extensive or expansive biofilm growth. And this could increase fecal indicator bacteria capture, but the effects on survival are less well known, and once again are likely to be linked to the actual constituents of that biofilm. So those are shown in particles B and sort of dashed particle B. And then the final one, sort of related to predation, we know that moisture can increase the mobility and the abundance of protozoa in soil systems, which could increase grazing pressure. So that's sort of our little green particle C, which look like they've been swallowed by little gray hedgehogs. Those are my illustrations of protozoa. Not very artistically talented in that regard. But we also know that at some point, it's going to be too moist even for the soil protozoa. So that's going to have a limit at some point. The final thing I want to point out here, sort of moving on to slide 14, is that all three of these biofilter components can impact hydraulic residence time distributions. So I've shown an actual biofilter residence time distribution from a Parker et al. Submitted manuscript, so it's not published yet. And you've got residence time in minutes on the x-axis and salt concentration, a conservative tracer concentration, on the y-axis. And I've got little arrows linking each of the different biofilter compartments to the residence time distribution. So for our vegetated compartment, you could imagine that roots are going to promote macropore formation. And what that means is that you can have roots that grow down into the biofilter, then maybe they die back a little bit, forming a preferential flow path in that system that could result in having lower residence times in your system. On the sort of filter media side of things, your grain size distribution matters. You could have a high percentage of fines, for instance, in your system, and that could increase the residence time in the system if, as the water is moving sort of through those fines, you get more friction, for instance. And if you go look at the submerged zone, well, one of these things that the submerged zone does is it creates sort of a enhanced moisture in those dry periods between storms. So you get reduced fissure formation and less cracking, so fewer preferential flow paths, which could, increase the residence time in the system. Similarly, biofilm formation, which we just talked about, could also increase the residence time in the system. So really the take-home here is that the effects of individual biofilter components on fecal indicator bacteria removal can be direct, which is what we were talking about in sort of the three previous slides, or indirect through changes to hydraulic residence time distributions. So moving on to slide 15, this means that we can sort of create a simple conceptual model for fecal indicator bacteria removal by biofilters, where just we say that multiple different biofilter design components have the capacity to impact fecal indicator bacteria removal, things like filter media, vegetation, and submerged zones, and acknowledge that the effects of these components may be direct or indirect through changing hydraulic residence time. So what I'd like to ask now, sort of on slide 16, is whether or not there are any specific components of biofilters or sets of components that are logical engineering design targets for improved fecal indicator bacteria removal. And I'm going to argue that in order to address this question, we need to evaluate the effects of biofilter design on fecal indicator bacteria removal holistically, looking across all components to avoid masking or misattributing effects. So how are we going to do this? Well, I think the first step is to compile primary literature on fecal indicator bacteria removal by biofilters, performing a kind of meta-analysis, and then using that data, perform some kind of path analysis using something like structural equation models in order to identify optimal networks of design elements that favor fecal indicator bacteria removal. And I'll talk a little bit more about structural equation models in a bit. But regarding the primary literature, sort of the meta-analysis, some of the key pieces of information that we want to make sure we have are things like log 10 fecal indicator bacteria removal values, vegetation presence and type, things like grasses, sedges, shrubs, at a very simple level, filter media percent fines, submerged zone presence absence, and infiltration rate, which I'm using as a mediocre proxy for flow effects, because we don't often have information about the hydraulic residence time in these systems, and so sometimes the best we can do is infiltration rate. So now I'd like to talk a little bit about the meta-analysis that we performed. This is on slide 18, and this is unpublished information, so I can't give you a citation for it. But basically what we did is we did a web of knowledge and Google Scholar search, looking for the terms biofilter, bioinfiltration, biofiltration, bioretention, or rain garden, and then fecal indicator bacteria, E. coli, Enterococcus, or coliform. And this found about 107 publications. And then we read the abstracts of all of these different publications, and to determine whether or not they were relevant, if they provided information on all the different metrics we wanted, and to make sure that they weren't duplicate studies reporting on the same data. And that left us with a surprisingly small number of studies. It's about 13 biofilter studies from across the globe, though it covered about 350 different biofilter runs. So there's a lot of data nested within those studies, luckily. 5 of these were from Australia, 7 from the United States, including Texas, North Carolina, and Maryland, and 1 from Israel. Regarding sort of the spatial scope of these studies, 5 of them were evaluating small biofilter mesocosms in a greenhouse or a lab, and 8 were looking at more permanent outdoor biofilters or large field mesocosms. And it's important to point out that the biofilter design varied widely across studies, because of course in order to look at the effects of different sort of design arrangements, we need to have that variability. Regarding plants, there were between 0 and 12 species planted in these systems, including grasses, sedges, and shrubs. Filter media composition ranged from between 3 and 46% fine sediment, and the drainage configurations could either have submerged zones present or absent. So once we have sort of our data from our meta-analysis, the next step on slide 19 is to perform a path analysis using structural equation models. So structural equation models are really just a statistical modeling method for evaluating networks of relationships amongst model components, in this case our biofilter design elements, where we use maximum likelihood statistics to estimate relationship strength. So I'm showing on the right sort of a schematic of our structural equation model of our biofilter, where we have our different biofilter design elements. So we have plant type, grasses, sedges, and shrubs. Filter media is indicated as filter media percent fines, and then we have submerged zone presence absence, all of which can impact log 10 fecal indicator bacteria removal. And one of the really great things about structural equation models that differentiates them from things like multiple linear regression is that you can evaluate both direct and indirect pathways. So here we also have infiltration rate in our model, and all of our different elements can either impact FIB removal directly or indirectly through changing infiltration rate. And on the left here, I have a couple of arrows, and I'm just pointing this out because it's going to be carried through for the rest of these structural equation model slides. When I'm talking about significant effects, they're going to be indicated with colored arrows, green for positive, red for negative, and big and bold for strong effects. And I'll also, to indicate sort of the significance of those effects, often give you standardized coefficients, and those are reported on a 0 to 1 scale, where 1 is the strongest it can be, and 0 is no relationship. All right. So moving on to slide 20, when we perform this structural equation models analysis, what biofilter design elements do we find contribute most significantly to fecal indicator bacteria removal? Well, I think the key point I need to make is that plant type, fines, and submerged zones all impact fecal indicator bacteria removal. However, submerged zone presence matters the most, and that's both direct and positive effects on fecal indicator bacteria removal and indirect positive effects where the submerged zone is decreasing infiltration rate and therefore increasing fecal indicator bacteria removal. Regarding filter media percent fines, we see only a direct effect, and it's positive on fecal indicator bacteria removal, whereas in contrast, the shrubs and grasses, sort of two of our plant types, are only impacting removal indirectly through changing infiltration rate. And interestingly, the effect of shrubs is to decrease infiltration rate, therefore increasing fecal indicator bacteria removal, whereas grasses are actually doing the opposite. They're increasing infiltration rate and therefore decreasing fecal indicator bacteria removal. An important thing I need to point out is that the model is only explaining about 32% of our total variance. This could be because, at heart, these are biological systems, so there's a lot of variability underlying these linkages. However, it's also possible and likely that we're missing model components. And a really likely one, of course, is climate. So if we move on to slide 21, I want to point out that climate matters. Indeed, if we include antecedent dry period, so that period between storms, the length of that period, as a moderating variable in our structural equation model, which basically allows the model to take on different configurations when the antecedent dry period is less than 10 days, so a wet climate or wet season, versus when it is greater than or equal to 10 days, so a drier climate or a dry season. And if you look at sort of our wet climate, wet season model, we see that fecal indicator bacteria removal is impacted most by the infiltration rate. So any reduction in the infiltration rate is going to increase fecal indicator bacteria removal. Whereas if we move into drier climates, we see that the submerged zone presence-absence is what matters most, and the presence has a positive impact on fecal indicator bacteria removal. Infiltration rate matters much less in that case. So the take-home message of all of this for me, and I'm going to give you a couple of these, this is sort of the first one, is that submerged zones, vegetation type, and percent fines, and infiltration rate are all important design elements impacting fecal indicator bacteria removal. However, submerged zones and infiltration rate matter the most. Submerged zones in dry climates and infiltration rate in wet climates. Then you've got plant selection, particularly shrubs, which were consistently decreasing infiltration rate, thereby increasing fecal indicator bacteria removal. And then filter media percent fines, which had by and large, sort of across all our models, a consistent positive effect on fecal indicator bacteria removal. And sort of looking at slide 22, if we want to sort of move away from models and take a sort of peek at the data itself, and we categorize it relative to those processes that were deemed most important. So we have submerged zone presence-absence on the x-axis, the log of the infiltration rate on the y-axis, and the log of the antecedent dry period, that sort of climate variable on the z-axis, or vertical axis. And the other thing I need to point out is that the color of each of these points is the log 10 FIB removal value. So we're going from a very high removal, greater than 3 log reduction in the red, to actual leaching out of the system, the blue colors on the bottom. And it's clear when you look at this that the highest bacteria removals, sort of where I've circled in the red oval, are systems that have submerged zones. They have moderate to low infiltration rates, and they have actually a long antecedent dry period, interestingly enough. Whereas if you want those systems that are doing poorly for fecal indicator bacteria removal, and of course we don't want them, but those are those systems that don't have a submerged zone. They have really high infiltration rates, and they also have that long antecedent dry period. Of course, this is not a perfect relationship. There are outliers. In fact, I've circled one of the ones there in blue just to show that there are exceptions to the rule. Another thing that I want to point out here, because I think it's really important, sort of moving on to slide 23, is while it might be really tempting to sort of look at this data and say, okay, we want systems that have really low infiltration rate, because that's going to really boost our fecal indicator bacteria removal, we need to keep other biofilter functions in mind when we're optimizing for fecal indicator bacteria removal to ensure that the trade-offs that we wind up with are acceptable. Because if we push that infiltration rate to really low infiltration rates, really what's going to happen in practice is that we're going to have a lot of water overflowing and going into the storm drain. We have poor volume control. So essentially we're just not treating that water, which is actually worse than the other alternative. So these are things we need to think about carefully. So take-home message 2, sort of moving on to slide 24, is that although we have successfully identified some key biofilter components that are logical engineering design targets for improved bacterial removal, uncertainties regarding why and how those features improve removal still remain. And to sort of illustrate this, I put the kind of biofilter model we were just playing with on the top, and behind that we have all of this complexity, and it's really a simplified version of the possible complexity space that we could be dealing with, looking at things like filter media amendments, protozoa and grazing, soil microbiome, root architecture, root exudates, and all of this, that, and the other. And I would argue that we need to understand the mechanisms that underlie the performance of individual biofilter components in order to further advance biofilter design. And currently there are some really great studies that are looking at characterizing some of this complexity, looking at filter media amendments, the soil microbiome, and invertebrate macrofauna in particular. But I feel that we need to remember, sort of as we're continuing to move into these more, I guess, novel spaces, that we also, as we're reporting sort of our novel findings, need to also report some of the traditional things that we were originally reporting, so that we can still keep looking at the system as a whole, sort of evaluating how all these compartment effects affect each other. And that way we don't wind up with some extremely good but disconnected studies in each of these areas that we can't consolidate into a holistic evaluation of biofilter performance and the things that we really need to target to improve our sort of our removal capacity and the effectiveness of these systems in the urban environment. So with that, I'm going to conclude my talk. At the back here, I have a set of references, just a really brief set, actually, that talk a little bit more about fecal indicator bacteria and stormwater, the environment, and more broadly, fecal indicator bacteria removal mechanisms by biofilters. If you're interested in diving into this further, that's sort of slide 26. And then the final 2 slides of the talk are actually just in case anybody wants to do more than a conceptual view of sort of the effects of those individual design components on FIB removal. These look at things a little bit more quantitatively, going through sort of a probability distribution and bootstrapped approach to show which individual components have significant effects, and they actually give, you know, the actual log reduction values. So if you're interested in that, you can look at slides 27 and 28. So at that point, I think I'm done, and thanks very much. I'm happy to take any questions.
Okay, thanks very much, Beck. Yeah, any questions?
Yeah, that was a very nice talk. What are your thoughts then on, so my take-home message from this as well is that you think allelopathy from plants is probably not a major role based on your analysis, is that right? And it's really, the effect of plants, you know, from what I understand you said, is basically driven through its effect on infiltration rate. So it's really physical effects rather than biological effects.
I would say that that's in part a limitation of the kind of analysis I was able to do. I don't know that anybody has really well characterized the effects of plants on fecal indicator bacteria in these systems. I know they're Anna Deletic and her group are going to be doing more work in that direction, but because the kinds of data that I could collect for a meta-analysis that are common across all those studies are more focused on flow effects, that may be part of why we see those as emerging. So I wouldn't necessarily rule them out, but I think it's an area that's important to look at more.
No, but a priori I'm a bit skeptical of that.
And they may or may not, I don't know.
I can see the attraction of the idea, but I'm not convinced. But anyway, yeah, I thought that was interesting. Yeah. And also, just one final comment. I really liked, you know, it's not directly related to the material, but I did like your point about undertaking studies which are sort of parsimonious to one another, or not parsimonious, probably complementary to one another, rather than, you know, this is one of the things of science, is it drives you to everyone to do something extremely novel, and at the same time, it sort of means that, in a way, you're not quite pulling in the same direction. So you know, it doesn't lend itself well to this sort of, you know, the nice meta-analyses. But then when you go to publish a study which doesn't have that extremely novel hook, then you'll get smashed. but it could actually be very important because it's providing a very important piece of the bigger picture puzzle. I think that's a good conundrum about the current system that we operate under.
Thank you. Any other questions from anybody?
So I have to ask a question about the plants, of course.
Sure, yeah, please do.
It's kind of surprising, not whether they did or didn't have an effect, but sort of the 3 different types that you found. So at least in your structure equation model, no effects from sedges. Whereas when, you know, for the other sorts of removals, sedges are usually you know, an effective plant type.
Yeah, that's been an interesting sort of conundrum when people have been doing these studies and they look at the performance of Carex in particular. Carex does not seem to improve removal of fecal indicator bacteria, but it's great for nitrogen, great for phosphorus. So, yeah, I don't necessarily know fully why that is, but that does seem to be true. Though there, in the wet system model, there was a direct effect of sedges on removal, and a negative effect actually. So they weren't helping. But so they can do something, but it's really unclear at this point exactly what they're doing.
Yeah, I mean, I like the way you went through the possible mechanisms. And I think the problem is we don't actually understand what the plants are doing. So we don't know what they're doing for the nutrients or for the metals or anything, and so we don't really understand what they'd be doing for the bacteria for the most part. Although you talked about, like for roots, I mean, if they're big roots, they can, you know, we definitely understand some sort of changes in physical, but not very much about the other ways they could be acting.
And I wouldn't be at all surprised if many of their effects are through the microbiome, sort of the native soil microbiome, and effects and interactions between fecal indicator bacteria and that microbiome.
Right.
And I know that there's a big sort of microbiome project that is currently being started at Monash. So hopefully they'll provide some insight in that regard.
Yeah, okay, that could be useful.
I'll put on my engineering hat too and say that the other wrinkle here may be a timescale effect. So typically we would measure removal over a shorter timescale. We went to Monash and ran some biofilter experiments in which we added sewage and looked at the removal rate. And so there's an initial removal that occurs that we're measuring, and that could be more physically based. Right, and we in fact find that the plants actually reduce that physically based removal because they create fast-moving water, the sedges anyway, which essentially reduce the residence time of the water in the biofilter.
Right.
On the other hand, you have a longer time scale, which we may not be measuring, which is what happens to those attached bacteria over time, which has to do with really the microbial community that's present and its interaction with the plants. And so the plants could have a really adverse effect on fecal indicator bacteria over those longer time scales by promoting a diverse community of organisms that are consuming them, for example, whereas over a shorter time scale could actually, the plants could reduce filtration, for example.
And that's one of the arguments for the submerged zone effect in the long term, is that you're essentially holding more of the water than you would have otherwise in the system. And that includes the fecal indicator bacteria associated with that, so more time to die, essentially.
Although, you also talked about regrowth possibilities, and at least in marine, the Enterococcus is potential, especially for regrowth. Did you pay any? So you're lumping all FIB together here. Did you look at? Or
I did parse them out. The problem being that I wouldn't trust at this point the data for enterococci because most people have been focusing on E. coli, given that it's more of a freshwater emphasis.
Right.
So it's kind of unclear to me at this point whether enterococci are removed more or less by these systems and how it all shakes out.
Yeah. Okay. Interesting.